Hi, and welcome to another edition of Ask the Expert at TELUS Science Museum. A big thank you to our sponsors of this event, Century Bank of Georgia, Marriott Courtyard of Cartersville, and Homelight. Their support helps us continue to bring you great events and content just like this. A few upcoming events to mention for you to put on your calendars. Our next Lunch and Learn on February 16th will feature Christine Walke, the Director of Education and Outreach for Southeastern Cave Conservancy. She'll be talking about caves in Georgia and how to explore caves safely, which is really important because caves can be a really dangerous environment if you don't know what you're doing. If you like caves, make sure to tune in on that one. On February 25th, for our Sci-Fi Night, our speaker will be Dr. Ed Album, who's the Interim Director of Agnes Scott's Bradley Observatory. He'll be sharing some of his fascinating photographs of planets, meteors, and other celestial objects taken from his observatory, and he'll also be talking about some good tips for taking good astrophotography. I want to mention also that February is our Member Appreciation Month. Check out our webpage for more details about the discounts and special opportunities just for members that we're going to have all month long. Today I'm excited that Dr. Penelope Boston is joining us to talk about her research as an astrobiologist and how caves on Earth might have something to tell us about other planets. She's the Associate Director for Science Development at NASA Ames Research Center. She has over 40 years of experience as an astrobiologist and geomicrobiologist and more than 25 years exploring the geomicrobiology and biomineralogy of caves. And she'll be talking about and telling us all about what those are today. In her distinguished career, she served as director of the NASA Astrobiology Institute, associate director of National Cave and Karst Research Institute, professor and chair of the Earth and Environmental Sciences Department at New Mexico Tech, along with receiving many awards and recognition for her achievements. Her research focus is geomicrobiology and astrobiology in extreme environments, such as caves, the geological processes creating caves on other planets and moons, and the use of robotics and other technologies to assist exploration in extreme Earth and extraterrestrial environments. So she'll be sharing some of her research with us today and her experiences. Welcome and thanks for joining us today.
Thanks very much. I'm really delighted to be here and be able to chat about some of the work that we've done in my research teams over the years. And we've had the privilege of going to many, many different environments. And so what I have done is put together a large number of images to really sort of give folks a flavor of what these amazing underground places are like. Let me share my screen and hope that is coming up. Let's see. PowerPoint. Okay. Here's my PowerPoint. There we go. All right. Can you see my screen?
Yep, we can see your screen.
Yeah. Almost there. All right. So that's a picture of me on the right-hand side of your screen in a fairly extreme outfit. And the reason that I have...
Actually, I don't want to interrupt you, but we can't actually see your PowerPoint. All we see is your screen with your list of...
Yeah. Okay. It should be showing it. I thought I was sharing, and the only way you can share. So let me try that again.
Okay.
Stop sharing. Open the share tray. There we go. All right. Can you see it?
There we go. That's much better.
Teams is not my friend, and we are forced to use Teams. It has its little
quirks.
Yeah. Okay. Is that good?
That's great.
All right. Okay. We're off on the journey. So the right-hand side of your screen, now you can see it, is a picture of me looking my glamorous best in this suit that is protecting me from a very dangerous environment. I am not by nature a daredevil. I don't seek out daredevil things to do for the heck of it. But the nature of the work that we have done really has taken us into a lot of places where, significant protection is actually necessary. Make sure I can forward. So, my general area of research has centered around geomicrobiology. That is really where the microorganisms in and on our planet interact with geological processes and geological materials. And for the past 25 years of my career, which is about not quite half of it, actually it's more than half of it, yes, I'm old now, has been done in subsurface environments, primarily caves. So I was not a caver. It was not a recreational caver when I started working in caves. It was really the science potential that lured me there. And the science potential is really great in the subsurface. It's an area where the microbial biodiversity is extreme, where many organisms have developed fantastic metabolic processes that we are not familiar with in organisms that we've studied on the surface. Microorganisms act as geological agents. They actually weather rock and mobilize minerals and do all sorts of cool stuff. And then, of course, the punchline for me has always been these things that we call biosignatures. A biosignature can be something as obvious as a giant dinosaur bone. We know from the studies of paleontology that such features are, the result of life in the past. So those biosignatures can also be very, very subtle, like a slight change in a mineral that indicates that microorganisms have been working on them. And so we bring that idea of biosignatures to the field of astrobiology, which is the search for the potential for and ways to study life beyond our own planet of origin. We're still in the very early days of this science. We have not yet detected life on another planet. But because we understand more and more about the planets, not only in our own solar system, but also the thousands and thousands, undoubtedly billions and billions of exoplanets that circle other stars in our galaxy and beyond, the chances for life having arisen elsewhere is really very high. So my joke always is that the work I do is an unparalleled opportunity to look glamorous at all times. These are some of the protective gear that we have used in various different environments. These environments can range from very hot to very cold to filled with poisonous, to us, gases. Microbes love those gases and they are living off them. But for us, it's a hostile environment, to protecting our skin against extreme acid conditions, protecting ourselves against extremely high levels of metals or high salt levels. And so that's why I wind up in all of this protective gear, is because we are seeking the organisms that are pushing up against the limits of life here on Earth. And that is for its own sake, because we're interested in those organisms and the processes that biology has invented and developed here on Earth, but also we apply those as models, as templates for what to look for on other planets. So if you wanted to sum up the human significance of this geomicrobiology stuff that I do, it really crops up everywhere. It's becoming clearer and clearer that even a lot of the economic minerals that we mine and use are only precipitated in the geological environment because of the presence of particular kinds of microorganisms. They certainly control a lot of what goes on in producing unusual minerals and crystalline forms. People are looking at microorganism properties and capabilities to help us clean up mining waste and byproduct materials that may be toxic in mining efforts. They engage in really weathering the rocks of our planet, and that puts materials into circulation. We know that right now we're having difficulty with our activities putting excess carbon into our system. So potentially microorganisms have the capability of reversing some of those processes that we are changing by our industrial and daily activities. Also, the temperature being high or low, organisms that can function in those temperatures. And the property of catalysis, which is the ability to make chemical reactions go faster, better, or happen at all, is something that biology does very well. And the organisms that grow in these extreme environments are capable of doing things that we are not capable of, but we can use their talents in industrial and pharmaceutical processes. Even when it comes to cave art and surface artifact of human civilization, like cave art or Egyptian pyramids or monuments, microorganisms can actually be damaging to those. And so geomicrobiology also has an applied field of looking at how we can make the conditions such that we can control any destruction of those precious remnants of our previous human activities. I use the scientific method, we all do, but when you work in a field like mine with field sites like caves that have not been explored, it's a very messy process. So, you know, we all learned about the scientific method in school. Some of us practice it, but that's a logical construct. What we actually do in reality is follow trails of serendipity, follow pathways that look interesting. And so it's a fairly complex process to take the kind of exploration that we do and turn it into rigorous science. I want to turn for a moment to the whole issue of extraterrestrial caves. So I have studied life in caves on Earth, but a lot of the application for this is, what about caves on other planets? When I first started doing this work, we did not know that there were caves on other planets. And when I first started writing about it, there was a lot of skepticism that there would be such things. But we now understand that there are volcanically created caves, not only here on Earth, but on a number of bodies in our solar system. And furthermore, any planet with a surface or a moon with a surface will develop cracks, either from external impacts like impacting asteroids or meteorites, or from motions, geological motions on the interior of that planet. This has led us to think about the potential for developing caves from entirely non-Earth mechanisms, meaning processes that we don't even see on Earth. And so I'm only going to spend very, very tiny amount of time on this because this is a whole other talk. But it's very clear now that we have at least lava tube caves on many different bodies in the solar system. This image collage shows some of these linear features where there are collapsed lava tubes. So you see the exposed tube, but where you see the red circle in the lower left, that's an area where it has not collapsed, and that is an extant cave. So there are giant pits, like you can see in Mars, the big hole in the center right. This is a gigantic 300-foot diameter pit crater cave on Mars. We now know there's about 2,500 such features on the planet. Even Venus, as hot and murky as it is, shows evidence of linear features that appear to be collapsed cave features. And even Io, which is the extremely volcanically active moon around the planet Jupiter, also has these weird high sulfur yellow lavas, and it produces lava tubes as well. So features are quite common in our solar system, and many more of them to be discovered. One of the things that we've learned about cave environments on Earth that really was an eye-opener to me, and I did not know before I started to become acquainted with caves and start working in them, is that the caves provide a window into the subsurface of Earth that is typically radically different from the surface. There are many wonderful caves in Georgia where your institution is located, and I have several friends who are very active explorers in Georgia caves, mapping them, looking after their conservation, and so it's very important terrain in many different parts of the world. When you go to a show cave, you very often come away with the idea that these are extremely rare, but actually the subsurface of Earth contains many cavities. And these cavities, we call them caves typically when they have a natural entrance, but it's probably less than 10 or maybe even 5% of all the caves that are in the subsurface of Earth that actually have natural openings. So perhaps 90 to 95% of all of that underground real estate is discovered.
So actually, I would just jump in really quick because we've got a question from Jason. You said that biosignatures could be as subtle as a change in a mineral due to an organism working on it. Can one tell the difference between a change due to a microorganism versus a change due to something else in the environment? And then the follow-up question is, how can you do that and what are you looking for?
It's very subtle. Digging for dinosaur bones, you either find them or you don't, and then you have to interpret them. With the microbial realm, it's a much smaller scale. It's much subtler chemistry. And the way that we're beginning to piece this together is by looking at what we find in natural environments, isolating the microorganisms that we find in those environments. And then one approach is the approach that I take, and that is to try to reproduce some of those mineral transformations actually in the laboratory, and then test that back and forth between the natural environment and the laboratory environment. It's very demanding to try to prove that there is no non-biological chemical way of producing each and every possible biosignature. So it's a very challenging thing, and it really relies on the preponderance of the evidence. There's no single silver bullet that says, oh yes, this is clearly microbially produced. It takes a lot of different lines of evidence to actually solidly make that claim. So that's an extremely excellent question. Thank you. So the difference between the surface and the subsurface is really illustrated in this set of pictures on this slide. The background is the very dry sand desert known as the Rub al-Khali in a number of different Middle Eastern countries. Two of my caving colleagues and friends, Susie and John Pint, have done a great deal of work in this environment. And I love to use this as an example because this area has been a desert for probably on the order of 20 million years. It's a very old desert system. And yet, you can see Susie peeking down into a hole on the lower left. And you can see in the center bottom, you can see a tiny caver repelling into a very big hole. Both the big holes and the little holes have caves that contain actual diveable pools. And in those pools, even though it's so dry on the surface, are remnants of the biology that lived in this environment when this was a much wetter environment on the order of 20 million years earlier. And this is true for both the microorganisms and the higher organisms that are found here. So even in caves with actual openings to the surface, the subsurface and surface boundary layer or boundary conditions can be really quite striking. And so the subsurface of Earth is a repository of biodiversity that does not exist on the surface, or in many cases does not exist on the surface. So why are cave environments so extraordinary? Well, number one thing is no sunlight for most of the cave, except, right at the entrance. And this shows the entrance little entrance drop in Leche Villa Cave, which is in New Mexico. They typically have high humidity, even in deserts, like I just showed you. The temperatures within a cave are very constant. They only will vary maybe a degree or less, other than the immediate influence of the entrance over very large distances. But they range all over, the average temperature ranges all over the entire planet. So you can find hot caves, you can find cold caves, you can find caves that are geothermally influenced and hot and steamy. When you're an organism, that constancy of temperature is actually a benefit. And the high humidity is a benefit because you don't have to protect yourself against the drying of day and night and seasonal cycles and arid times. The challenge is that nutrients, that is food, is really pretty rare. So if you're an organism that needs to eat organics, which certainly you and I do, it's a pretty sparse environment. There is organic material that is washed into caves or comes in to closed cavities by way of fractures in the rock that carry groundwater. But the organisms that are the most extraordinary and the focus a lot of my work are the ones that essentially are rock eaters. These are known by the technical term as chemolithoautotrophs, which just means they use chemicals from rocks to feed themselves. And the process is similar to photosynthesis in that they are fixing carbon dioxide as biomass, as sugars and biomass. But instead of using the sunlight for energy, they're using chemical transformations that they cause in the mineral environment in which they live. So it's a pretty wild out there branch of life. The truth be known, they are much older in terms of those mechanisms than the kinds of metabolisms that organisms like us use. But because they are mineral consumers and they work in this geological setting, that means that is the foundation for them to alter the geology and mineralogy that produces these subtle biosignatures. One of the wonderful things about caves is that they don't have surface weather. They have internal weather-like processes, but to such a small extent compared to the surface that you get wonderful preservation. And we see this not only these subtle biosignatures that I'm talking about, but also ancient animals and plants that have fallen into caves, a lot of archaeological evidence of humans and artifacts. And so they're museums of their own sort. They're sort of auto museums that can preserve materials amazingly well. So if you've been to most of the tourist caves in our country and even around the world, you will know that they're made out of limestone, and they often have these beautiful secondary calcite, which is the crystalline form of limestone, features, speleothems, as they're called, like stalactites and stalagmites and many more. But the truth is that caves occur in just about every rock type you can think of. And I'm not going to slavishly go through these. I hope that your eyes have time to roam around these, but there's everything on here from caves in giant rock salt deposits in Chile, all the way to caves in Antarctica made out of the Antarctic ice mass itself. And so the geochemistry that is the background for all of these amazing microorganisms that use rocks and minerals for food is an intrinsic part of the cave story. And this is where a lot of the extreme variability, the biodiversity, and the amazing visual appearance of a lot of these biosignatures occur. So if you're a microorganism, you are very tiny. But you don't live alone. You live with a lot of friends. You live with a lot of friends of your own species or strain, but you also live with many, many different other kinds of microorganisms. And microorganisms really in nature function in communities, almost indissolubly. So the work that they do on rocks and minerals is very often this orchestral arrangement of microbial talents rather than just looking at a single strain. And so this is challenging to work with in the laboratory because classical microbiology has been based on the medical model for good reasons, which is trying to find, a pathogenic microorganism that might cause disease in humans or animals or plants of significance to us. But the truth is, in nature, they behave in these great aggregates. And that's the way that they function in the cave environments. So there are unique cave formations that are very extraordinary. Here's just a potpourri of 3 that we have worked on. On the left hand, you can see my colleague, Mike Spilby, taking what looks like white mud off the surface in a sample. This white mud is the product of trillions and jillions and gazillions of microorganisms that are busy essentially disaggregating, almost melting, taking apart the limestone bedrock. And they're doing this because they're mining for tiny, tiny quantities of organic materials. And then by mobilizing that mineral, it reprecipitates out in the stuff that's called moon milk. That's an old translation of a German term because people used to think it had something to do with the moon for reasons I don't understand. But it's this white fluffy stuff, but it's actually a microbial product. So even though the organisms are tiny, the caves can be liberally coated with this stuff. If you look at the upper right hand, you can see these pool fingers. These are sort of like hangy-downy stalactites, but they're not stalactites. They form in clusters from the edges of pools. And what we have found is when you slice them across and look at them in detail, you find that there are episodes of coatings that are entirely non-biological. And you can tell that by the crystalline structure and several things about several properties of the carbon involved. And then that's interspersed with layers where microorganisms are involved, where we even find the microfossils of them. And so those are clearly also a microbial product. The cave pearls that occur in a lot of caves around the world are literally like pearls in that every one of those spheres that you see in the lower right-hand picture is actually loose within its nest. So cave pearls, form separated from the little nests in the calcite in which they form. And this has always been a subject of great curiosity, but what is going on there is that the microorganisms are producing sticky gooey stuff. We call it biofilm. What you brush off your teeth every morning is biofilm, right? So microbes produce this all over the world in all different kinds of environments. And that keeps the minerals depositing both on the inside of the cave pearl and externally in the nests. It's a very curious process.
So actually I have another quick question to throw in there. So is it the same kind of microbes in every cave that has moon milk, or is it different microbes working in different ways, or?
It's we originally thought that it would be the same microbes, but what it is the microbes with the same talents. And so these same opportunities for microbes to make a living occur in caves all over the world, and yet the microorganisms very often differ greatly. In some cases, they're genetically related to each other distantly, and in some cases, these capabilities have arisen in very different groups. So yes, excellent question. Thank you. Lechuguilla Cave is a famous cave. It's in New Mexico. I point to a caver. You can see the caver head and shoulders and upper chest on that lower left. So you can see these glorious chandeliers, we call them. These are made out of the mineral gypsum. When it's in this crystalline form, it's called selenite. But gypsum is what wallboard is made out of in your house and buildings. So it's a very common mineral, but in these caves, it produces these amazing features. There's over 150 miles of map passage, maybe I think it's 170 now, since I've made this last updated this slide. It's 150 feet at the deep point, so it's the deepest cave in North America, and it's massive and extraordinary. It was actually chewed out of the limestone bedrock by sulfuric acid dissolution instead of the other mechanism for forming caves in limestone, which is weak carbonic acid in rainwater and groundwater. So there's huge chambers and amazing mineralogy, vast rooms, intricate mazes that you can barely make your way through, and it is filled with dozens of different kinds of microbial communities that are acting on the rocks and minerals. One of the most striking ones is this iron and manganese fluff. We call it speleosol. So a sol is the term for a soil, and speleo is the term for caves. So this is truly soil that is being made inside the cave by microbial activity, breaking down the limestone and releasing a lot of the iron and manganese trace materials that are in the limestone. And this appears in the limestone as a chemically reduced form. And so the microorganisms are taking that chemically reduced form and oxidizing it into a more oxidized form. So they're changing the fundamental isotopic character. And by doing that, they're getting these tiny quanta of energy, and that's what they're coupling to CO2 fixation. As we talked about with photosynthesis, this is chemosynthesis. And as they do this, they work their way down through the rock, and they leave all of this byproduct for us to find. So we took about 15 years of work before we felt really confident that we were seeing a true microbial byproduct. We've now reproduced this in the lab in all different ways. Some of these microorganisms are extraordinary, and in fact, in caves, most of the microorganisms are previously unknown to science. So the biodiversity is just mind-boggling. We don't even really know how to deal with it yet. These are electron microscope pictures that show some of these amazing forms. One of the forms that a lot of these cave microbes take are star-shaped. And if you're familiar with, most surface microbiology, the forms of most microorganisms are fairly simple. They're either spheres or rod-like forms or, little spirals. But in the subsurface, there is a huge diversity of form. And part of this is because the microorganisms, I think, are not being grazed by as many higher life forms. So it's almost like you took a trip back in time to a point before higher life forms had arisen on this planet. In many of these caves, especially the most chemically extreme ones, there's very little or nothing in the way of multicellular organisms. And so it's a microbially dominated world. They are the kings and queens, rulers of their empire. And so they produce these amazing forms and bizarre filaments and the fluffy stars that you see here. These are some of the experiments that we have done to really try to figure out what these organisms are doing. And the green pictures that you see with bright spots on them, these are fluorescence stained rock cores drilled into the cave surface, into this what we call punk rock, which is the rock that's being broken down by the microorganisms. And those bright spots are the cell heads of the organisms, and the little filaments are filaments that they put out into the rock in order to look for materials that they're looking for. Some of them are extraordinary, like the beads on a string, pearls on a string that you can see in the set of images on the right. And we have been finding these for many years in caves all over the world. It took us 20 years before we finally were able to grow them in the lab. Finding them in nature and growing them in the lab are two very different things. And one of the things that is a biosignature is where I've pointed to, called tiny dino footprints. And here you can see that these beads on a string separate and they attach with these little sort of flat holdfasts, these little footprints onto rock. And when they ping off, it leaves a little bathtub ring where that was. So even in the absence of the organisms, you can look with an electron microscope at certain rocks and see that the microbes had been present. So we've reproduced all this in the lab. I show here just quickly a couple of laboratory agar plates, and you can see the dark spots. These are the organisms that are busy producing this iron and manganese fluff, and we've induced them to do this in the lab. So that, back to the the previous person's question, that is closing the logical loop between their capabilities to do this in the laboratory and the verification of the fact that that's probably what's going on in nature. Sometimes we produce magnificent crystals, and so you can see the Switzerite, this beautiful manganese iron phosphate mineral in the upper image that doesn't get produced in the absence of the microorganisms. So just having dead organisms in the environment doesn't work. They have to be alive and working. And they'll produce dark patches like you can see on the agar plate in the middle, and then gradually that becomes more and more crystalline. It goes through the whole classical sequence of minerals that is understood in mineralogy. But if you kill the organisms and continue to let it incubate, that crystallization doesn't go farther. I want to take you now to Mexico for a couple of minutes. This is in the state of Tabasco. And the cave is Cueva de Villaluz, the cave of the lighted house. And this is because this cave has a number of different skylights that open to the outside. It is a relatively small cave. It's only a couple of miles of map passage. And this is because this is an active sulfuric acid cave. So it is forming and breaking down as it's being formed because the sulfuric acid is so aggressive against the limestone. So this cave can never become very big because as it's made, it is, crumbling essentially. But the remarkable thing about it is, even though this is so poisonous to us that we have to wear breathing gear and skin protection, it is a tremendously biologically rich system. And it's really the hydrogen sulfide gas that is coming in from a number of different sources, both geological and biological, that is driving this system to great acidity. There are beautiful cave-adapted fish. There are all sorts of insects. For reasons that I do not understand well yet, none of us do, bat species are able to live in this toxic environment. All of those little dark specks that you see in the pool, those are all the fish that are adapted to this environment, and it is all driven by the microbiology. So if you look here, you don't need to pay attention to all of the data on the slide, but you can see us in the suits in the center. We're gloved up and we are wearing protective breathing gear to keep the hydrogen sulfide from our lungs. The phlegm ball mats in the center, those were discovered perhaps about 20 years ago as we were just poking around in the sediments. The walls have these acid organisms called actinomycetes growing on them. There's both biological and chemical precipitation of elemental sulfur. So it's a real stew of an environment, and it's all being driven by the energy that is contained in the hydrogen sulfide gas, the H2S. And the base of the food chain is the microorganisms splitting that H2S, getting the energy from the hydrogen that it splits off, and as a byproduct producing elemental sulfur and many other sulfur materials.
So we have another question for you. So this kind of, I think this relates to as you're using the caves as a proxy, or Earth caves as a proxy for space. So Michael, who's 9 years old, asked, how will astronomers ever find aliens?
How will astronomers ever find, well, we're trying in a lot of different ways. So astronomers are working on a very different area of astrobiology than I do. Astronomers take telescopes and spectroscopes, and they look at the chemistry and physics of distant planets and try to figure out whether there are world-size biosignatures. So an example of a world-size biosignature right here on Earth is the oxygen that we breathe. So we breathe oxygen, and that oxygen is produced by the photosynthetic organisms on our planet, like plants and algae in the oceans. And we would not have free floating gaseous oxygen to breathe if it were not for those biological processes. So our Earth has this magnificent oxygen global scale biosignature. And so that's the kind of scale and type of thing that astronomers are looking for. So the most famous weird features in this Cueva de Villaluz sulfuric acid cave are these Snoptites. And you can see why they're called Snoptites, because they basically look like what comes out of your 2-year-old's nose when she has a cold, right? Chemically, it's not that dissimilar. So they're gooey, they're slimy, and they're made by a bunch of different kinds of bacteria. But all of those bacteria and archaea, they're related microorganisms called archaea, they all produce sulfuric acid, and they produce this protective slimy stuff. The pictures on the lower left are electron microscope pictures showing these tiny microorganisms on this gooey stuff up close. And you can see that I have to be gloved and masked and all of that in order to collect this kind of material. So this is an amazing system. And we think that this kind of activity was actually what was going on 7 or 8 million years ago in Lechuguilla Cave that I showed you before, when Lechuguilla Cave had no natural openings and was way, way deeper than it is today. So we're trying to connect up ancient biological activity in caves and where we can find modern biological activity in caves. So we had these drippy, gooey things that looked like they might be biologically produced in Lechuguilla Cave in New Mexico. And this picture is from quite a long time ago, in the the mid-1990s. And we did analyses on this stuff, and we could not find any biosignatures. The only biological thing about that rock formation was that it looked biological, that it was curved, and we couldn't figure out any way that the mineralogy could do that by itself. It wasn't until years later when we started to study Cueva de Villaluz that we got the clue that yes, what we were seeing was a mineralization transformation of these sulfuric acid gooey things that are still growing right today in Tabasco. We also find these gooey structures in other sulfur caves around the world, but even some non-sulfur chemistry environments produce these. So even things like old submarine depots in Crimea in the Ukraine, we have found these, not a sulfur atmosphere. And so clearly the microorganisms are the foundation of these kinds of potential biosignatures. We also find things that look like wiggly squiggles, little hieroglyphic type patterns. And it turns out that those are also biologically created, that the organisms are essentially spacing themselves on vertical and overhead walls. You can see a close-up in the lower left of these sort of little bumps. These are bumps made out of gooey stuff that microorganisms are making that also trap particles of clay and also allow minerals to be precipitated actually there. And when you stain them with a fluorescent stain that looks at only live organisms, you can see this blue staining on the lower right. So we know that those kinds of patterns are also microbial action. If you go into lava tubes in many different parts of the world, you will often see what looks like shining glitter on the walls. And the shining glitter is really because what looks like mineral patches, like this pink and this white silvery patches that I show here, are actually a combination of microorganisms that have a complicated life cycle and the minerals that they're reprecipitating on their surface. And these complex life cycle organisms actually become water repelling for part of their life cycle. They become what's known as hydrophobic or water phobic. And so that means that the high atmospheric water vapor level in the cave will condense on them rather than wetting them. And so it shines like a million diamonds in all of these different amazing colors. And if you see that, you have the chance to go into a lava tube cave and you see that kind of shining pattern, you know that you're looking at a vast metropolitan area of microorganisms. Some of them are quite spectacular in shape. So the electron micrograph in the lower left-hand side, I call it our chrysanthemum garden. These are beautiful microbes. The diameter of one of those little fuzzy guys is probably about 1/100th the diameter of one of your hairs. So these are minuscule little structures, and yet they're so elegant and beautiful. And these tend to be very common in the lava tubes. Here's a few more pictures from lava tubes around the world. The picture on the upper right shows all these different patches of color. And if you look at them, they just simply look like deposits of different kinds of minerals. But here again, water is beating up on them. And this was our first clue that these were actually microbial communities of different types, all growing on this lava surface in Hawaii. We've studied them in the Azorean Islands, in Hawaii, Mojave Desert in California, all over the world where there are lava structures that we can study. I want to end with this. This is perhaps one of our most spectacular environments. This, when it was first emailed to me, some pictures from this site in Chihuahua, the state of Chihuahua in Mexico, I thought it was a joke. You know, the caving community is a global community. We sort of all know each other, and there's a lot of joking and you know, prankstering and things of that sort. And I literally thought when I was first sent a picture of this that it was a Photoshop joke. But it turned out not to be a joke. And in 2008, 2009, we had 2 National Geographic sponsored expeditions into this environment. You can see that people are in these orange suits, and these crystals look almost like they're ice, but they're not. This system is extremely hot. So in Fahrenheit, this is on the order of 130 to 150 degrees Fahrenheit. It's hotter than most saunas. And so those orange suits that we're in are actually not to keep us warm, but to keep us cool, because we're packed in ice vests. We have ice packs underneath our caving helmets, and we have backpacks that are actually actively cooling our breathing air, which is flowing over ice that is in the backpacks. So an extremely demanding environment. One of the most amazing things about these giant crystals, of course, is that they reached this huge size. The only reason we could get into this system is really because of mining. So these caves had no natural openings. They were discovered accidentally as a byproduct of mining for lead and silver and zinc and things of that sort. And as part of the mining process, they came across these. And because mining requires you to get rid of the water in a mine, they're always pumping the water out. If it's below the water table, which is the case here. And so these crystals actually grow in these caves entirely fluid filled. So it's only the mining that operations allowed us to get in there. These areas have now been reflooded. Whether or not they'll ever be drained again, we don't know. But we had this one precious, slice in time to actually get in there and study what's going on. So these giant selenite crystals, once again, the mineral gypsum in the selenite crystalline form, has little pockets in it. And the small piece down the bottom, you can see one of these little so-called inclusions, these little holes, natural holes that form in the crystals. And within those crystals, there's both solid and fluid materials, and that's what we were really interested in. I originally wanted to just see if we could find any kind of sequenceable DNA left over, but it turns out that we actually were able to find live organisms trapped within these little space capsules, so to speak. And the walls themselves that are not in the crystals, all those orange walls that you see behind, are actually all also studded with microorganisms. There's also fossil materials. You can see the pictures on the right-hand side are all fossil materials. Let me just end up with one more picture. I want to end with this, which is what looks like a mesh stocking. We call them microchoyas because I live in New Mexico, and we have these cacti called choya. And when they die, they have this skeleton of woody material left behind that looks just like this, except those are big cacti. And here again, these are microscopic. So one of those little mesh stockings that you see is perhaps the length of it is maybe a third of the diameter of one of your hairs, and the width of those little tubes is once again about 1/100th of the diameter of one of your hairs. We don't know what these are. We have found these all over the world in caves. We have hunted up one side and down the other. We have tried to identify them. We have done all sorts of DNA studies. We have done microscopy. I have not yet been able to grow them. We're virtually certain there's something alive, and yet trying to find them has been elusive, and we've been working on it for a very long time. So even here on Earth, we essentially have what I think of as native aliens that we're still trying to figure out. And so, that just illustrates the challenge that we face as we try to investigate life on other planets and moons in our own solar system. And as the young man was asking before, trying to investigate the traces of life, elsewhere in our galaxy around other stars. So let me end there and thank everybody for showing up.
Well, thank you so much. We have just a couple more questions for you. So Jason has a question. For things like Mars rovers, are we looking for cave openings and the ability to go into any extraterrestrial cave and do any kind of analysis? Or are we looking for similar types of formations? I think he means like here on Earth that have the same kind of structures.
Yes, so space missions to caves are incredibly challenging. I've been working on various concepts for that, as have a few other people, for a very long time. And there is, finally, after all this time, there's really some serious attention to this. It really requires a very different robotic approach than we have seen with current missions, where we have wheeled rovers and so forth. I've been involved in a number of different projects with roboticists. We're trying to develop climbing robots, tumbling robots, robots that bounce, all different ways for getting into these difficult environments. So that work is coming along. We're not ready to send a mission yet. I sure hope that, sometime within the next 10 to 15 years that we will be able to start sending reconnaissance missions into caves. Certainly our earliest opportunity is going to be on the moon, where there are many pits and lava tubes. And so even exploring those for usable ices for our human astronauts probably will be the first areas that we're able to actually investigate. the moon is not thought to be a place where life has been able to occur. So our best astrobiology goal is either Mars or it's some of the icy moons around other gas giant planets in the outer solar system. From my point of view, when you have an ice-covered ocean moon, to me that's just a fluid-filled cave, a fluid-filled ice cave, and the way to go about exploring it is much more similar than it is to exploring an ocean.
So Karen has a question: is Jacob's Well a limestone cave?
I don't know. I don't know what Jacob's Well is, but if that's in Georgia, yes. Stone cave.
Okay, and we've got a follow-up question, our last question for Michael. He actually wants to be an astronaut. Do you have any tips on becoming an astronaut?
Become very good at your chosen field. So more and more we're choosing astronauts not just as test pilots, although we still have plenty of those, but the latest crop of astronauts, I did quite a bit of training with them. Astronauts get trained in all different kinds of science and engineering and operations. And so I trained the last astronaut group in astrobiology. And they come with all different kinds of credentials and talents. Some of them are scientists. Many of them are some kind of engineer. Many of them are medical professionals. So, science, engineering, math and analysis, computer skills, all of those technical fields are fair game for astronaut talents. The critical thing is that to become an astronaut is a relatively long process, and it's very, very competitive. But it's competitive in a way that really draws you into becoming the best you can be at the fields that really matter to you. So if you're passionate about medicine, follow that, and then turn your attention to the astronaut corps. If you are passionate about being a test pilot, become fabulous at that, and then you can apply like that, or become a scientist or an engineer, and you can apply from that stance. So I wish you well. I think there are going to be increasing numbers of astronauts required, so that's the good news for people interested in that line of work. It started off with a few people back 50, 60 years ago, and the call for astronauts is coming online, and we also have commercial enterprises that are looking for astronauts. So I think it's a growth field. So good luck with it.
Well, thank you so much for taking the time to talk with us today. It was very interesting, and I've learned a lot myself. And I'm sure everyone who's tuned in has learned a lot as well. So thank you very much.
Pleasure. It was great to talk to you guys.
Thank you.
Bye-bye.